Hello, everyone. Welcome to BrainMap. This seminar series is co-sponsored by the Fee-funded Center for Mesoscale Mapping housed in the Martinos Center. It is our great pleasure today to have Dr. Joseph Mandeville for giving us BrainMap talk. Joseph Mandeville is an assistant professor of radiology at Harvard Medical School and assistant physics at MGH Martinos Center for Biomedical Imaging. After obtaining doctorate in physics, he held the position of research fellow at Martinos, Massachusetts General Hospital, prior to joining the professional staff. A primary focus during his career at MGH has been characterization, optimization, and interpretation of neuroimaging signals. Early work included the development and characterization of contrast-enhanced FMR, which is now the fMRI tool of choice in clinical models at most field strengths. Recent work has focused upon ways to improve measurement and analysis for PET studies of occupancy assisted by FMR. I would like to remind our audience to address your question at the end of the talk and by raising your hand online. The stage is yours.
Okay, thanks for the introduction. So if you're, I just want to assure you that I'm not going to talk about the whole everything to do with PET detection, analysis, and interpretation. You know, if you go to a conference like neuroreceptor mapping, you can take full courses on tracer kinetic mapping and maybe acquisition. We're going to focus on a subset of the studies that I've been involved in with dopaminergic systems since we acquired the PET MR scanner, almost all using raclopride. I'd like to upfront acknowledge a lot of the collaborators, especially Christine, Sandra, and Monica that do a lot of the NHP studies with. Iris and Bruce have been longtime collaborators with its PHMI going way back. And you know, there's a whole list of people who have contributed from our center, from Copenhagen, who we had a collaboration for a while, from Catholic University in Leuven in Belgium, Wim van Duffel and John Arsenault, and early in their studies from the Gordon Center. So let's start with a series of questions. So we get this new machine, and you know, we came from fMRI world, and so one question we wanted to ask is, can we use this to better interpret PHMRI studies? Can we make PET more dynamic? You know, from the fMRI world, you look at PET and it just looks like a slow old dinosaur. Can we make, you know, why are so many radio tracers insensitive to agonists? There's a whole world of agonist radio tracers that just don't function like we really want them to, right? If you give them an agonist, they just don't move. An antagonist will displace anything, but agonists won't. But some of them work well, and so you know, why is that? Can we really use PET to study natural task activity? You know, prior to getting this, there were already reviews. I'm using raclopride for natural task activity, like playing a video game in the scanner. Oh, we see something. Can we believe that? Can we actually do that? And can we combine PET fMRI to produce new biomarkers? Because, you know, whenever we get a new tool, we always say, let's get new biomarkers. So let's, you know, start with some questions. And, you know, up front, we've got to acknowledge some PET limitations too. There aren't enough PET radio tracers. It's a frustration for everybody in the field. And some of them, as I said, don't work well for displacement studies. Worse, it takes, you know, some talented PET radiochemists to produce this. And if you don't have the bandwidth, you can't get them, even if they exist. I mean, you have to fund these, and it's difficult. And you know, PET is expensive, so these are, you know, problems we have to work around. And as I said, so in this, we won't be talking about a whole series of radio tracers. We'll be talking about one of the most well-characterized ones, raclopride. All right, so this is sort of an outline of the talk. Let's start with the background, which is PHMRI and PET. And you probably thought that I'd start later on, but let's start way back, right? So, you know, before I came here, these guys started functional brain imaging. And one thing that was cool back then, you don't get it from these mid-career pictures, everybody in the center was young, right? So the first time R01 now is over age 40. So by that standard, everybody was young. Jack was young, Ken was young, and you know, Jack had this, you know, method where he used contrast agent, and people kind of poo-pooed it as like, well, you have to use contrast agent. That sort of, you know, preceded what we use now all the time in animal models. And, you know, we changed, you know, long-lived contrast agents, but there's more than one way to skin a cat. And we use that because it works better in animal models. Ken came along and it had this electrifying result that you don't need contrast agent at all. And I wasn't there because I wasn't in the field yet, but my understanding is like the first 10 talks next year at ISMRM were all counter-rotating flashing checkerboards and look, oh, we see bold in V1 too. And pretty soon the field progressed. And I do the second one because, you know, quickly they went from sensory to perception and cognition. And you can do a lot of things that you can't do with the PHMRI that we're doing. You can look at faces versus objects. You can scramble the objects and do all the control tests. But within 5 years, suddenly NIDOs are coming to us and saying, okay, now what can fMRI do for drugs? Right? As PET has a proven role, but let's give fMRI a chance. And it's just a totally different animal. So Bruce and Iris kind of in tongue-in-cheek named it PHMRI, which is could still pronounce fMRI, right? But there's real differences, right? It's a direct neurochemical intervention. It has temporal response, which now reflects neural activity. You don't have to convolve it by any hemodynamic response because it's so slow. It's low frequency. It's often refractory. That means there's less averaging. You get one shot, so it really puts a premium on inherent sensitivity. And you use different analysis method. And the truth is, we didn't really know what to do when it came. And so the initial progress is really slow, and we basically had to figure out how can we actually detect anything. So that's why we started doing the contrast-based imaging. We weren't even sure, like, you know, there's even controversy about what questions to ask. Should you be looking at the direct neurochemical effects? Should you be looking at the, you know, the how it's perceived by the subject? And, you know, we were doing things like, you know, correlating it with 2DG and with, you know, CFOS, which is an immediate early detector signatures, which weren't very, I think, appropriate. And, you know, Bruce came along and started, you know, doing systematic studies, viewing it as just another readout like cyclic AMP. And we could look at, in some sense, you could look at, you know, the direct coupling and create sort of models of this. And so now I think that we have a direct functional readout like FDG. It works for all drugs, unlike PET. And we're learning how to better interpret the signal, but it's tricky because it has some weaknesses, right? It doesn't tell us anything about drug targets or occupancies. It's sometimes corrupted by non-neural events. Sometimes it tells us too much. I mean, if someone's in the scanner spacing out and enjoying it, I mean, you're seeing a lot of things which may not be directly related. So PET can be useful in many ways, right? So take a quick step back. Now PET, you know, we need a radio tracer, right? So we look at something kind of like back projection. We then analyze it with multi-compartmental models. We can then create maps in space. And if you image long enough, you need a certain amount of time to define a binding potential. If you get the precision down well enough, you can, before it decays, you can use the rest of the time to find a change in binding potential. And the ratio of the change to the resting state binding potential gives you the occupancy. And that's what we refer to as drug occupancy. So when I talk about occupancy, that's it. And just a side note, most PET studies report steady state measurements. So, you know, this is actually a small slice of the PET world. So, okay, we got this PET-MR center. We started wondering, what can we do? You know, we were used to thinking about flow and metabolism. We spent all this time thinking about this. And FPET has taught us, you know, maybe a better way to use PET to look at metabolism. But for PHMRI, the more interesting thing is occupancy versus function, because you can, you know, look at what's actually driving it and connect it with what we want to connect it with. And if you want to make models about how that connects to function, that's fine. But you don't really need to, you know, metabolism really doesn't really tell you that much more than PHMRI. You really want occupancy and function, right? So this thing arrives around 2011. We don't know what to do with it. We start thinking, okay, we'll do occupancy versus function for the drug. And then, you know, later it's like, do we actually do this for a task? Well, let's start with DVS first as an intermediate, right? So the first thing we wanted to do was start with simple selective agonist and antagonist. So this is kind of like sort of, you know, a tour of what we were doing for the last, you know, sort of a decade, right? So the first thing is that we created a receptor-based model. And this was actually useful because it you know, kind of integrated a lot of the things that we've been thinking about, like why do we see biphasic responses at some dopamine levels? We see, you know, negative responses at low levels and positive responses at high levels. We see differences across species. We can all put this into a model, and then we can go and try to verify some of that models, some aspects of that model with PET. Unfortunately, can't verify all aspects of that model because, you know, like I said, if you want to see like the occupancy at D1 receptor, but D1 doesn't tell you occupancy. That radio tracer just doesn't work that way. So we can use fMRI to inform PET, right? So, anatomy is a really useful thing. Motion correction, people are, using it, of course, for other kinds of PET corrections. You can use it for reference for functional localization, like we'll talk about the DBS, or we can use them together, both to characterize drugs, this is an agonist, a partial agonist, etc., or to provide novel markers like internalization. And so my background, like I said, was in fMRI or pH MRI. And so early on, we started seeing, you give something which is relatively selective, you get all these interesting responses. You can get biphasic responses. Like, why am I getting these biphasic responses? Something that should be hitting a single receptor. And so, one of the things that kind of lumped onto was maybe this is, you're seeing aspects of dynamic receptor regulation because, there's this literature that you hit something with an agonist and get internalization. And so I don't know if that was going on. That's the story we had. We went with it. But now we can start to investigate that. So just a background, internalization is this, sort of, in a classical model, we like to think all the receptors are sitting there waiting to be hit. But if you hit them, there's a homeostatic process that says, I'm going to run in high, and slip inside the membrane. And it's just a dynamic homeostatic method to downregulate response in the face of overwhelming neurotransmitter, right? And there are beta-arrestin knockout models that we can use to have the blunted internalization and desensitization. And there's also potential implications for interpretation and disease, like schizophrenic. Some people have hypothesized, well, if it's hyperdopaminergic, maybe that's a dysfunction with downregulation due to dysfunction with receptor trafficking. So it's possible, right? So how do we interpret occupancy? Just getting back to the math, we could have a binding potential. Binding potential is composed of three things. You usually think of it as a concentration of receptors, but it's not that. Part of it is masked by how many are bound by the basal occupancy. So we never see that unless you want to go through sort of a Frankenstein experiment where we actually deplete the dopamine from T-brain subjects, which people have done, but you don't want to do it very often. But there's also an affinity there, which is the ratio of on to off. How well does it bind, and that affinity can shift also by things like receptor trafficking. So we have these three things. And we have this experimental problem, which may be related to these, which is that some radio tracers, you give an agonist, you have no measurable effect. You can give like a whopping dose of agonist to NNC 112, and you almost see just nothing. Some of the early PET literature, there were paradoxical increases. There were radio tracers like spiperone. You give it, and reliably, the binding potential goes in the wrong way. And suddenly it was a surprise when Rapid came along. It's like, hey, this is doing what we expect to do, and it's displaceable. And so, there's a sort of, ideally, we could just work with the classical model, but that's not realistic for any radio tracers. It's, it's not the fault of PET, it's the fault of biology, right? That these receptors are changing. And some of them work well, like raclopride, PHNO, MTA. There's diprenorphin works poorly, but carfentanil works better. And so we want to at least see if we can address this with a radio tracer which is well understood. So what can we actually see? So I went back and looked at a 2011 poster before we did all of this, and we were doing complicated studies. We had this awake monkey. This shows a awake monkey in the scanner getting FDG. We were thinking about these complicated, receptor trafficking. So this is, we're thinking about already models of internalization before we've done any of the studies. And we really need to simplify things, right? So we got together with, a committee of the Martinos Intelligentsia, and they said, why don't you start with something simpler? And so, which was a really good idea. So we started with a series of studies led by Christine Saunder, and one was the antagonist. And the antagonist is just much easier to understand. And that's probably how you want to start if you're trying to look at PET fMRI coupling. So that was a really good idea. But if you look at these in space, they're quite similar, right? You could, in both cases, as you give more drug, you're displacing, you're knocking out the PET signal, you're increasing occupancy. At the same time, you're inducing fMRI signal. D2 is inhibitory, so if you give it an antagonist, you inhibit inhibitory, so you get excitation, With the agonist, you get the opposite. You can plot this, peak CBV versus occupancy, so only a couple animals, but everything looks fine, right? Now you look in the temporal regime, and like I said, the antagonist is working great. But the agonist, there's this funny thing where, well, we're expecting this long response of the PET, like you see this PET, right? This is a model, and you can't really see how long it is, but the fMRI is just over in a flash, right? And there may be, at a small level, there may be some other things you don't see, because you lose, as time goes on, you lose some things in drift corrections, right? But the major effect is over quick. And so there's an agonist. So we went back to the models. We've been thinking about this before doing the studies. And, if there's no internalization, if the internalization time constant is infinite, there's nice coupling. And that's kind of what you get for antagonists. Because antagonists, if you're looking in vitro, they just don't do much for internalization or externalization. And as you start to increase that internalization content, right, you prolong the PET signal, probably because it's going to internalize, that shifts the affinity stays blunted, even after, for long after the neurotransmitter has gone away. But for fMRI, once it goes inside, the neurotransmitter can't cross that cell membrane, it can't access it. And so it abbreviates the fMRI signal to go in the opposite directions, and you keep playing the game, and you try to match the signal, and you say, maybe this is fast, it's like 5 minutes. And it kind of matches the in vitro internalization time constant. So success, right? So how else can we look at this, right? That's nice, but can we use a more a targeted way? And we have knockout mice. So there's a pre-existing study by Skinsberg et al., I guess from Robert Innes at the NIH. It's a nice study, and they look at the effect of genotype versus time for MMPA and for Falipride. And so this is basically a delayed phase imaging where you give it, and then you assume that at some point in time, based upon literature results, the dopamine has gone away, and if it's internalized, an animal without internalization, the binding potentials recover, and if it's internalized, it should stay depressed. But these things are happening in parallel, right? So the receptor, the neurotransmitter is going away, receptor trafficking is occurring and maybe externalizing. It's just a very subtle effect. Surprisingly subtle, right? But they so we actually saw a decrease. So here, it depends, right? So it's somewhat similar. So they saw similar, right? So this is wild type and knockout in the baseline studies. In our study, I'll show you that we got somewhat less in the knockouts. Little counterintuitive. So we thought, how else can we probe receptor tracking? Well, okay, we could do the same thing, and we were going to different radio tracer. But maybe we can try to imagine a more sensitive way to do it. And the more sensitive way is, if we could do a second challenge. And the thing is, let's imagine that there's no shift in affinity when receptors get internalized. In that case, there's just binding potential is just completely insensitive. It's not like a door. The membrane is not like a door, it's like a window, right? It just goes in and out. There's no difference between the inside and outside. And so there should be no difference between binding potential should be completely insensitive to internalization, but occupancy should still be able to see it. And that's because we know that the neurotransmitter can't cross the cell membrane, right? So because of that, it doesn't matter if the radio tracing can get it. We should still see a pool of receptors that can't be bound by dopamine. Yes.
You want to remove this, or is it okay? Because it kind of covers your. May I remove it?
Sure.
It's kind of.
Oh, sorry.
No problem. I thought you might want it there, but
I wasn't even looking. No,
yeah, I apologize.
No.
I should have done this in the beginning. And if I may, I will also. I'm sorry. This is easy.
Okay. All right.
Sorry.
So we can sort of do a model, and if there's no affinity shift, there's no effect on binding potential, but there is a big effect on occupancy. And if you have twofold affinity reduction, they get a little closer, but still occupancy is more sensitive. All right. So here's a study design. So you take wild type and knockout mice. You either pre-treat them or you don't. And 3 hours later, you come along and you give a within scan challenge. And so we did 26 wild type, 24 knockout, divided into 2 arms, one to measure binding potential, one to measure occupancy. And to address sort of the high cost and variance, we want to do multi-mouse studies, right? So we create a mouse hotel. So we do 6 at a time. And you multiplex the anesthesia and two floors. And it's a tricky study because I couldn't do this. I need help from Jonah, our former tech, was amazing. Every mouse needs 2 tail veins, right? Because you have to have one. You can't inject a challenge through the same line as the radio tracer, or else you end up decrypting an experiment with flushing the radio tracer. So six mice at a time, 2 tail veins per mouse. So the boar had all these catheters coming out of it. We had to create even a catheter holder, right? So it's kind of a mess, right? But and then another problem is you have a Harderian glands, which are these funny things in the eyes of rodents that apparently exist to suck up every pet radio tracer ever created. and the effects kind of bleed into striatum, and that's not a problem for most of the studies. But when we pre-treat and you're already knocked down the binding potential in the striatum, now the striatum is starting to much lower binding potential, and now the fact that you have this contamination into it becomes a problem. So you have to do a partial volume correction. So we went to the, geometric transfer matrix formalism. But also, I like to see these things. So we created another formalism, basically said, let's take GTM, but instead of relying on anatomical regions, we use a B-spline basis set. And so we can perform sort of simultaneous partial volume correction and smoothing. And now you can see in the post-amphetamine, it goes from this top right down to here, and you can actually see you've restored the binding potential. So, okay, you can see that this partial volume correction is working. All right, so you apply this, and this is the results. And to answer, Bruce's, the binding potential is a little bit different here, a little bit lower for knockout animals. And, because, these things aren't exactly well matched, you have to use ANOVA, and you go through and you look for binding potential, the change in binding potential. And for each one of these little colored rectangles, it illustrates the effect size. So a knockout mouse has a slightly lower binding potential than a wild type mouse. So the knockout effect is here. The effect of amphetamine pre-treatment is easy to see, right? But what's really interesting is the interaction between amphetamine and knockout. And that is significant also for both of these. And if you come back and normalize the results, you actually have a subtle effect on binding potential. But you have a really big effect on the change in binding potential. And if you look at, so you can think that the absolute change in binding potential and the occupancy. So the occupancy is just normalized by the baseline binding potential. In this case, in the knockout mice, which don't have internalization, occupancy is conserved, which feels kind of good, right? You come back to the second challenge and you're getting sort of the same thing. In the wild type, it's significantly blunted, right? So our hypothesis is sort of came to be true that that a second challenge is more sensitive. I don't want to rewind, but I just realized I didn't mention. I come back here. This is from Christine's study, and there is a second quinprole challenge here. So with the fMRI, also goes away, right? A different model, but you're seeing the same sort of thing, right? So okay, so now we have extra confirmation using a knockout model. So so you can I want to make sure that I get to some of the rest. So okay, so now I'd like to we're thinking about like biomarkers. So one potential biomarker is internalization, but it takes a little different. It's a little difficult because you have to give if you were going to translate, you have to give it to a human subject. You have to give a strong dose of agonist, right? So then you have to have an fMRI technique which is sensitive enough so you can pick up this dissociation. The PET will be able to see it. And this is a case where the fMRI can see it in principle, but you have to do a lot of studies, right? And you probably have to use something like ASL rather than BOLT. But so we'll get to that a little bit. But so let's look at human translation of D2 agonist study. So we did a study in non-human primate, and one thing we noticed is that if you look at occupancy and percent CBV, you get different curves in the putamen and caudate, right? So if you think about a model for that, percent CBV relative to occupancy. So, oh, just give an antagonist. So this is with cold raclopride, right? So, and the idea is simple. It's if the antagonist really has no activity itself, what you're seeing is you're driving the dopamine off. If you drive it all off, it's a measure of occupancy, right? And to the extent that it's linear, the change in CBV is going to be a measure of the basal dopamine occupancy, and the change in binding potential. The maximum change in binding potential you get is 1 minus that, right? And so you have by taking this ratio to the extent that it's linear, you could get something which it reflects basal occupancy. Now it's not quite linear, and so we explain this curve as the fact that we give an antagonist. Actually, you're also raising the level of dopamine by binding the autoreceptor, and that goes out. And at the low occupancies, it doesn't matter because there's not much dopamine. At the high occupancies, it doesn't matter because you're swamping the system with antagonists. But the middle occupancies, it bends the curve because you have more dopamine competing with it, right? So for human studies, we needed a well-tolerated drug. And so one thing we decided to look at is what's out there. And one thing which is out there is a failed antipsychotic called prochlorperazine. It's used as an antiemetic. An antiemetic means you come out of surgery and they give you a painkiller, and they found out probably from off-label use that this worked. And so we thought, okay, this is basically a typical antipsychotic. We tried it in the monkeys, and it fell pretty much right on that curve. So these two diamonds up here are points that we tried in the monkeys. So let's try it with humans now, right? So this is visual validation that we're getting something. If you look at the cerebellum and look at the putamen, we're doing mostly bolus studies, and sometimes it's hard to see, but if we look at drug versus saline, see the saline washes out much faster, right? And so if we do modeling, we see, okay, by modeling says there's a big change in occupancy here, and almost nothing with saline. Probably, if you do enough studies, hopefully it would be consistent with Eric, right? We didn't want to do a lot of saline studies, but anyway, you need a method to measure function. And bold signal we knew wasn't going to work based upon all our studies in animal models. So we used PCASL. And this shows you it's noisy, but if averaging is your friend, right? And if you average a lot, you can get it. And so prochlorperazine was well tolerated by subjects. The PET binding potential was easily measurable. We did see putamen greater than caudate, just like we saw in the monkeys. So that's consistent with sort of the microdialysis literature. So maybe it's kind of a measure of basal occupancy. There are a lot of problems with the studies. This is one of the earliest studies, and we started on base 6 before we realized you can't really do human PET MRI on base 6 because halfway through the study, you have to turn off the fMRI, or you can't rely on the PET. Right? It's just a low count problem on that scanner. So then we switched to the other scanners with some X2 scanners. We started off with bolus plus infusion, but it was one of the first raclopride studies. So we had to switch to bolus only because we couldn't get the yields. And we kept lowering the dose, but we didn't lower it enough. So there are a number of problems with the study. But one big problem is we just saw a much more nonlinear response. And so if we try to model this, so this is the response, and maybe we're seeing 3.5 times. And the problem with that is we're not simply measuring basal occupancy. We wanted to say this relationship is kind of linear, but it's not very linear, right? It's wildly nonlinear. So as a simple translation of a nice idea, sort of failed, but we still should publish it. But yeah, anyway, I wish we had more points at lower occupancies, but we kept lowering the dose and we still had relatively high occupancies. And that's interesting because every one of the subjects said it was well tolerated, but I did talk to an unnamed person in the center who said, I had an acute dystonic reaction. I was bouncing off the walls, and I had to go back in after they gave me compazine after a standard procedure. So one reason to publish it was just that they mostly switched to other antiemetics like ondansetron. You can give boatloads of that, but to the extent that they're still using this, maybe just knowing that occupancy is high, that'd be useful. Okay, so most have been talking about interpreting things at high levels of dopamine occupancy. Let's shift to the other extreme, which is low levels, localized dopamine release. So okay, we can detect, easily with strong drug. With raclopride, it doesn't matter whether it's agonist or antagonist. I mean, there's maybe we see the displacement longer than we expect with an agonist, but it works fine, basically. So how about a behavioral task? Well, one of our advantages of working with, a lot of talented collaborators is you have opportunities that don't normally come up. And one of those opportunities was working with WIM. They were doing deep brain stimulation of the ventral tegmentum. And so we decided to use that and use deep brain stimulation as a sort of intermediate model. And so what defines most detection limits? Well, motion artifacts. And you think, well, of course, motion artifacts, and that's, it's obvious, but I just want to say, I just want to point out just how insidious these are with PET. If you look at raclopride, all the binding potentials in the attainment, it doesn't matter which direction you move, it looks like displacement. Every movement in the world looks like, oh, I get dopamine release, right? And so so this is a study from Mike Levine, who worked with Cyprian, and simulated bias for motion, and you get this nice response. If you average across another studies, looks like, wow, I get nice uniform displacement throughout basal ganglia. But one of the real signals, it just low CNR. And one of the problems is you have a low signal to noise. So averaging across the whole study, the temporal signal to noise is like 10 if you're from a single voxel. And later in the study, it degrades because the radio tracers degrade. And later in the scan, maybe it's like 4. So you have to really aggressively smooth the data. And finally, you have small changes in binding potential. This is a study I took where they're looking at the difference between amphetamine and placebo. And they wanted, there may be real differences. Placebo may have real effects. But they want to say that, placebo and amphetamine have a lot of similarities. But if you look at these different regions, these are all regions of attainment. I would say there's no difference between any of them here. There's no difference between any of them here, right? In other words, there may be, the main thing is you're having a hard time distinguishing the effect of oral amphetamine. I think this was 0.15 or 0.25. So low dose oral amphetamine, it's really even hard to distinguish from placebo. So that shows you this could be even harder for a behavioral task. So, and finally, there's model limitations. So there's all these limitations, and we can address the signal to noise, I think, by some smoothing, small changes in occupancy. We would like to use agonist radio tracers, but like I said, got to use what's available. And we tried, but we weren't able to get an agonist radio tracer. And modeling, there's bias variance trade-offs, but we'll talk about it. So how to use compartmental models. So usually we use a reference tissue, and the reference tissue, we assume that there's uniform non-specific binding. And for robustness, we use a collapsed tissue by using a fast exchange approximation. And by collapsing, we do this. We take a model that says you go to the extravascular space, and then you have specific binding. And we say, you know what? We're just going to ignore all that. It just goes into one sort of space. And this is all, it becomes one model through a fast exchange approximation. And SRTM is robust and is accurate enough for most drug studies. And the thing about one of the good things, one of the bad things about reference tissue modeling is getting reasonable accuracy is super easy, right? Because it's most sensitive to the ratio of integrals. So if you look at SUV or relative SUV minus 1, which just means just fit the data past some time period, you get like something like this. If you look at MRTM2, but you ignore the most dynamic term, you get something that looks like this. If you look at the 2-parameter SRTM, you get this. And if you look at a full reference tissue model, you get this. And it's like by eye, they all kind of look similar. And so, if you look at plot effort versus accuracy, most of the accuracy comes at really low effort, and it works it's really hard to get better accuracy. I mean, you put in all this work to eke out this little extra, piece of accuracy, but maybe it's worth it. So here's the indication of model bias. The first indication we got was we look at these drugs, which weren't expected to have a huge effect. So this, for instance, is a D3 receptor antagonist, and D3 there's not a lot of D3. It's not it's not wide uniform throughout all of the basal ganglia. And we give it, and we get this response, and it doesn't look biological, right? And we saw this response numerous times. We saw it in one of Monica's studies with dipernorphin, which is generally considered not very displaceable radio tracer. whenever we try to see displacement, we squint hard enough, we get this study where this result where we see pair displacement in the middle surrounded by this paradoxical opposite effect, right? There's probably no result, because if you model this, you realize, okay, at high binding potential, we expect a positive bias, and at low binding potential, expect a negative bias. Kind of explains this result. And here I tried to put a single, hotspot here. But so this is a real result. All this stuff is artifact. All right, so this is how I think of SRTM. I basically think, the SRTM approximation, if you think about this, is the SRTM equation. And what's underlined in red is the missing part. And the missing part is this extra term, which is not in the typical model. And it depends upon a convolution between the rate of tissue change and this exponential function, which has a dependence on binding potential in K4. So the SRTM approximation is good if you get to a steady state tissue concentration. This goes away. So if you ignore all this and you do embolus and you can reliably get to this, SRTM is pretty good, right? It's good at large K4. In other words, if it's fast enough, it makes sense. If the binding potential is big enough, right? Problem is, if you want to use it across a distribution of binding potentials, it's going to be good some places and bad some other places. And if you give up this, you're giving up some of your signal, which is difficult, right? So onto the unilateral DBS. So this a study that John Arsenal and Wim started and published in Leuven, and then Christina was leading here. And John Arsenal, I feel bad. He was this kind of, he left the field, but he was actually instrumental. So he was not only did studies with Wim, but he came over and really started our studies and taught us how to do this. And it was a nice model. So you go into VTA, and that means that the area you try to detect has no artifact from probe, et cetera. It's a projection, right? And it's unilateral, it's titratable in principle. Still really hard. So if we think about dopamine function through fMRI, so this is sort of the way we're thinking about it. You get these dopamine pulses. You get a whole series of dopamine pulses. And fMRI is composed of a D1 term and a D2 and a D2 term, and they go in opposite directions, right? And oftentimes the D2 wins because your affinity for D2 is higher than your affinity for D1. But low affinity receptors are also more reactive, right? The high affinity, they go there, they stick in there, and then if you do a bunch of repeated stimuli, the low affinity ones work, right? Win out, right? So they each serve a role. So you're seeing the D1 work, and then Christine did some studies, which again, we haven't published, and probably would be great to publish, but this is a problem of having it in too few monkeys. But she did a D1 antagonist and D2 antagonist, and D1 antagonist reduced it. The response, D1 antagonist increased it. But you couldn't knock the whole thing down. And in other words, so we thought, I guess the conclusion of this, preliminary as it was, is that at least part of it is dopaminergic, but not all of it. And we could come along and do this in humans, but again, you've got to be willing to give large doses of antagonists. It'd be nice to be able to do this with PET, right? So the PET approach to DBS is that you have to do this in some sort of blocks form because the PET just doesn't have the temporal resolution. So we used within scans challenge repeated sessions in one non-human primate blocks. Our first analysis was sort of this typical MRTM2 plus challenge, and then we used this regularized full reference tissue model. And if we compare them, if you use the full reference tissue model, but you fix K4, we could have called it the simplified reference tissue model again, right? You're really simplifying it. It's just you're fixing the K4 to be a constant value, which is, 1 over K4 is non-zero. But otherwise, it's, you're fixing the value. And you're saying the value of this K4 is representative of the radial ligand, rather than it's just this fast exchange approximation. And because of that, it's not surprising that the reproducibility is similar. What is encouraging is the stability is better, which means that if you just truncate your data and say, when do I get the same binding potential that I would get early in the study? get it at much earlier time points if you use this approach than if you use the SRTM. So in other words, you can either do your studies earlier, or whenever you do your studies, you can assume that you're having less variance because you've converged on the binding potential sooner. So that's a win, right? So this is the best single session. So we look at fMRI p-values, it's lateralized. We look at the FRTM, the change in binding potential. Okay. And I show this because I question, can you see the displacement? You really can't. I mean, maybe you can see it, but this is the best. And this just shows you how difficult this is when you're getting down to these levels of displacement. Right? This is maybe 10%-ish, right? So below this is going to be hard. And so we average across a whole bunch of sessions with fMRI, and at a certain p-value, p-value threshold that makes sense. This is what fMRI looks like. This is what we getting the same thing here. This is what the MRTM2 looks like. The thing which survives is this negative surround. It's not quite as good as it looks, because actually, if you lower the threshold, we are getting lateralized, but it's not nearly as well lateralized as the fMRI. So, so why it works, and mostly why it works, is we're reducing the dependence of the change in binding potential on binding potential. And in the SRTM, your estimate of binding potential occupancy is heavily dependent on the baseline binding potential, and it becomes much less accurate at low binding potentials. And you have this variation, and you just can't pick out the real changes in the presence of this bias pattern. And we probably still have a bias pattern with the other. Okay, so there's a room for improvement, of course. I'll talk about that a little bit more. Okay, so attempts at modeling human dopamine release attacks activation. All right, so okay, this is hard, right? So we tried the delayed incentive monetary reward. So you give a prompt, so you take these, high valence faces. It's like, okay, you super lose, right? That's minus $8 just lost. Up to, okay, you just won $8. So you play this little task in the scanner. You do that because it's well known to activate basal ganglia, and it's been hypothesized that this is a dopaminergic task. It has elements of reward, has elements of motor planning, etc. So you look at during this monetary incentive delay period between, and then they respond to the task, and the outcome is you responded fast enough, and you adjusted the delay time. So if they're working their hardest, maybe 2/3 of the time, so they'll get it right, and 1/3 of the time they get it wrong, and they slowly work up to something like $200, and they go away happy, right? And with fMRI, we get pretty robust activation of basal ganglia, so we're working as we want. And if you average across these regions, you see that big gain and big loss are sort of similar, that they both give you kind of big activation. And probably because a lot of this is just due to things like motor planning, which have nothing to do with reward per se. If you look at specifically nucleus accumbens, you get a slight bias towards the big reward over the big loss, which kind of makes sense. This in both this study and in earlier studies by people like at I think it's UCLA who has done this, who was doing this quite a while ago. Okay, so PET, what to do about PET? All right, so well, we can't really take a region of interest approach. I think we need to use a voxel-based approach for something like this. So we try this, this geometric transfer matrix with B-splines. It's lots of basis functions, just kind of shows basis functions, a few basis functions across a mouse brain. Go up to human, you get like 26,000 across a three-dimensional field of view. And that means you have a matrix of 26,000 on the side, right? So these are big matrices. And why do we do this? And you do this because if you need to boost SNR, if you use Gaussian, you're blurring the binding potential. You're bringing white matter in, and you're mixing tissue types. And you can do better than Gaussian by using non-local means, which unfortunately has this I think unfortunate acronym now because it's mostly local, and it's it's basically signal-based smoothing, right? So instead of smoothing in space, you smooth in signal space, which means you smooth along signal contours. So you smooth along high binding potential regions or like binding potential regions, right? So selective smoothing, and you avoid some of the partial volume that way by near the edge, instead of blurring across that boundary, you smooth along the edge, right? So, and we can do better than that by using this B-spline GTM, where it's like Gaussian smoothing, but then you go back and you repartition the signal that you know you've lost based upon a geometric model of the blurring, right? And so, all right, so can we get displacement? Sure. If we cheat. So it's not really cheating, because there's no standard way in the literature to do this. So the standard method, if you want to use MRTM2, which is most people use, you calibrate the reference region outflow rate constant based upon a high binding region. How high is high? Up to you. So if we choose a binding potential region greater than 0.5, we can get displacement all throughout basal ganglia. And I chose 10 to the minus 4. We could raise this a little bit, but this would survive. This just shows you the change in binding potential at kind of a low threshold. And as we slowly lower this, it goes away, right? And all we're doing here is we're looking at if we do simulations, we're shifting these bias curves from being mostly positive, in this case, to being more negative, and the high binding potential barely positive, so it doesn't survive. And so we're kind of like playing with model bias, but it's also not well described in literature. Okay, there's other simplified approaches we can use. So one approach, I said, if we looked at SRTM, this is the problem. It's this uptake region. Now, I show this as a nice steady state here, but of course, anyone who's done PET studies know that if you have steady state in one region, you're not going to have steady state in another region, right? Because binding potentials vary. So you're always going to have some. So this has some benefit. So the method is basically ignore this uptake, and by ignoring it, you're basically going from ordinary to weighted least squares. You're also trading off bias, reducing your bias, but you're increasing your variance because you're throwing away some of the data. It doesn't work as well. It works well in some areas, not in other areas. So it has some benefit, but it's not great. Another maybe better way to do it is, instead of fixing this K2 prime, which is basically what MRTM2 does, we say, the 3-parameter model has somewhat better variance than the 2-parameter model. So let's just don't fix the value, let's fix the functionality. So we just measure the functionality versus binding potential. And you'll see the low binding potentials get progressively higher. It's a monotonic function. You fit that function. You actually haven't introduced any more voxelized variables. And so it should have the bias characteristics of the 3-parameter, but the variance characteristics of the 2-parameter. It should be a win. And we see that, more reactivation goes away, and it seems like the game is to get rid of all of our response, unfortunately. So as far as I can tell so far, we find methods equal smaller response. Other approaches, so I've tried this FRTM, and essentially it's given similar but not better, and probably slightly worse results than this sort of parameterized SRTM method that I just described. So basically, I guess the bottom line is no luck so far. So what would we expect? Let's go back to what would we expect. So cyclical voltammetry, people have actually done VTA stimulation in monkeys, looking at dopamine. They say if you do DBS stimulation, you get, they don't have a great calibration, but they're thinking about maybe a 2-fold increase in dopamine. And we're able to barely see this with DBS, but they say, juice reward, which is not exactly what we're doing, different, monkey, human, whatever. But they're saying it's 10 times smaller. Natural reward is 10 times smaller than DBS. When it's 10 times smaller, we don't have a prayer. fMRI comparison, we can also do that, right? So we have fMRI signal for human studies, bold, monkey studies, anesthetized, CBV. So we have to, kind of do some comparisons based upon earlier studies. If we do that, we'd come to the conclusion that maybe the DBS is 2.5 times bigger than them. So somewhere between, 2 or 3 times smaller natural reward versus DBS versus 10 times smaller. So it's definitely smaller. So it's much smaller, and I think, of course, it matters if we're ever gonna have a chance with that, because there are more games you can play. Going to higher SNR scanners, using agonist radio tracers. We haven't been able to do that, but those are things, it kind of matters where we lie on that. Additional model considerations, which I'm not going to really discuss, but you realize that there's a lot of things that's kind of swept under the rug, right? So gray and white matter are not the same. And if you're dealing with PET, partial volume is inescapable. Some people say, I don't care about the white matter. Well, you have to, because your scanner is giving you gray and white matter everywhere, and even in basal ganglia, and so you're having some sort of contamination with that. And if you look at just in the white matter, the kinetics are different, and that's been known for quite some time. And one possibility is a gray, is it your non-displaceable volume fraction is different. We just assume that everything is like the cerebellum. If you're using gray matter in the cerebellum, comparing it to white matter in some other region, gray matter has like a different free water fraction than white matter. White matter is more lipophilic. It's not clear that these things are the same at all. But possible interpretation problem is that, if we start playing around with the non-displaceable fraction, that's going to affect our inferences about binding potential. And it's going to increase our apparent binding potential in the white matter. And it may not be that we're looking at in other words, we may, the breakdown may be not just this, but there may be the failure of the 2-compartment model. In other words, if we're really going to compare gray matter and white matter, we need to consider the 3-compartment model, which says there's the extravascular space, the specific binding space, and the non-specific binding, right? So it's more it sticks differently in gray matter and white matter. Additional model consideration. I assume that K4 is like globally constant. So here's a map of K4 for racopride on this side and for PHNO on this side. And PHNO is a D3 preferring. And for racopride, we're fitting an extra parameter, and there's, that always brings up some variation, but it's remarkably consistent. It's not true for PHNO. And if you window this, you'll see that we're seeing this, these big K4 values in these regions, which are thought to have high values of a D3 receptor. So it may be that you're actually using this to actually tease apart parts of the binding of PHNO between D2 and D3, which is useful. So if we look at it, just PHNO binding potential versus K4, they're quite different maps if you threshold them. And so it could be useful that way. And more generally, there's been this long-standing concern in the literature that rebinding could introduce a functionality between K4 and binding potential. And so mostly this has come in the context of the fact that people see very different values for the offset or the K4 value in vivo and in vitro, like order of magnitude different. And they've tried to use this to explain the difference between in vitro and in vivo values. But that the in vivo values are so sensitive to other factors that for instance, there's like a factor of like 4 just raising it from room temperature to body temperature. So probably this doesn't describe that, but I thought I'd do a little test. So one way to think about this model first is we think about the measured K4 as proportional to the actual K4, but it tries to get away. And as it tries to get away, there's the probability of escape, which depends upon diffusion and et cetera. And there's a probability of it rebinding. And the rebinding probability, we kind of know, because that's a typical law of mass action, right? That's like the K on times B available. That's how we determine the binding potentials. And if you invert this, you get the 1 over K4 is just a linear function of binding potential. So we've got 1 over K4, we expect a linear function. This looks at, in these up here, this is, so ignore the red curve for a second. These two, I looked at 2 studies of in non-human primate. One was quinpro, one was racapide. We look at the same region, putamen. So you're not changing the tissue, same tissue. You look at one study, one full scan before the challenge, one full scan after the challenge. So they're dual scan studies with something in between to knock down the binding potential. You try to measure the K4, and you see that the K4 value in both values shifts to smaller values as would be predicted by this. So it could be that you are seeing this rebinding effect. So then I said, okay, now let's look at in humans we have no such study, but we can look at two regions with different binding potentials. And most putamen, it's kind of similar. We can look at putamen versus thalamus. And there we see sort of a similar effect. So it could be that, we have to incorporate something like this. So let's go back at the end with some of these questions. Can we better interpret PHMRI studies? I would say yes. That's one we made the most progress on. The very first thing that we did before we did any of the studies was develop this multi-receptor model of, fMRI response to dopaminergic stimuli. And it really described a lot of the things sort of quantitatively when seen versus dopamine function. Some of the biphasic responses to amphetamine at low doses, mid doses, high doses, etc. Other people have created Han and Gittiv, had progress looking at some of their serotonin questions. So here I think it's been useful, and I think it's limited, somewhat limited, because like I said, there's some things you can't study. Or you can study with the PHMRI, but PET doesn't provide much input. You want to study, glutamate. There's no displacement for glutamate. You want to study D1. There's no displacement for D1 radio tracer. Can we make PET more dynamic? Well, limited success. I mean, we haven't built any new scanners or anything. Cyprian's working on it. One thing we've done is, talk about limited. Trying to get success where you can by going to a slightly more accurate model where we can move challenges to earlier times, get more accuracy with the models at earlier times. Okay, that's some success in making it more dynamic. Why are so many radio tracers insensitive to agonists? I wish I could tell you. Basically, not much progress there. I mean, I think we have some added some insights into why radio tracers like racapide, which are sensitive to agonists, why they see this prolonged response. But and I want to say also, coming back, can we interpret PHMR studies? And just not going back to some of our early collaborators. I recently saw some data from Dustin Wooten, who you guys might remember, who's used to work with the Gordon Center and did some studies with us, and Mark Norman, and saying that NNC 112 is a partial agonist, right? And so they did that based upon looking at low dopamine, high dopamine responses, combining, using PET to verify that they were getting the same occupancies, right? So in the absence of PET, you'd have a hard time making that case. So, I think in some places we made some progress. Can we really use PET to say natural activity? Nope. It's not as far as I can see. And the thing is, we're getting 2x or 10x away. It kind of matters, right? And I can't tell you which of those it is. And can we combine PET fMRI to produce new biomarkers? Internalization, I think, is I think it would be hard to translate, but sort of success. The basal occupancy shows elements of basal occupancy, but also it didn't give us quite what we wanted. But so there's, take victories where you get them. Thanks.